Good morning and welcome back to the channel. It's always beautiful having you here with us as we discuss different concepts in science. This morning we are looking at the enrichment culture. Enrichment culture and what is enrichment culture? We find out that microorganisms, like we know, are ubiquitous in nature, meaning that they can be found everywhere. And due to this, we realize that so many microorganisms can be found in so many places. And so microorganisms naturally are in a competition with one another for resources. Wherever you find microorganisms, you find quite a number of them, and they're in this intense competition for resources. So because they're in this intense competition for resources, sometimes it can be difficult for some of the microorganisms to thrive, to do well in the presence of other microorganisms. Now, there's a man called Bejnik, who is a scientist who designed a method. The method or the idea behind the method was to enhance, improve conditions of nutrition and of environment to make sure that while you improve these conditions, the general idea is to favor the growth of particular organisms to the detriment of other organisms. So enrichment culture techniques, we can say, are those techniques that we use as microbiologists to be able to enhance the growth of particular organisms to the detriment of other types of organisms. And this is important. It's important because many a times we want to study a particular organism, want to take out time to understand the features, the science, the microbiology of particular organisms. And it might be quite difficult to do this if you are growing them in competition with other organisms. And so this is important. It's important for us to remember that it's about the environment and the nutrition, manipulating the environment and the nutrition in such a way that it favors the growth of particular organisms to the detriment of other organisms. And this is what is done in enrichment culture. Now, for enrichment culture, there are techniques that must be followed to be able to carry out all this process. First is sample collection. You must be able to collect your samples. So depending on whatever it is that you're trying to culture the microorganisms out of, it could be soil, could be water, could be air, could be human samples, could be agricultural samples, could be human waste, could be industrial samples, whatever it is, even from built environments, from offices, from schools, from toilets, wherever it is that these samples are going to be taken, samples have to be collected. And so there are techniques for the collection of these samples. Now, after the collection of samples, another thing that has to be done is have to have a situation where we can process these samples. Samples can be processed through many ways. Sometimes we use peptone water, sometimes we use membrane filtration techniques, sometimes we use, there are different types of techniques. We can use buffer, phosphate buffer solution. So there are many techniques that we use depending on literature, depending on what those before us have done. We can use these techniques to, prepare our samples. Now, after the samples have been prepared, then we want to create, we want to incubate these samples, we want to create defined conditions, conditions of temperature, conditions of pressure, conditions of pH, you know, conditions of nutrition. So we want to define, manipulate conditions in a way that these conditions favor the growth of these organisms. This is quite important. When we do that, we also want to monitor the growth of the organisms. It's important to monitor the growth of the organisms. This monitoring could be done, it could simply be something as simple as a color change or visibly seeing colonies being formed on the agars, on the culture medium. It could be change in color of the culture medium, whatever it is. We need to be able to determine if our medium is growing and then see the growth of our medium. And then lastly, we want to be able to isolate pure cultures. So there are subculturing techniques that we use to be able to isolate pure cultures of our from our samples. Now, there are different types of enrichment cultures, and it depends on what it is exactly that we are trying to do. You can have enrichment cultures, if, for an example, we are trying to culture out nitrosomonas, for an example, you need an enrichment culture that has a high nitrogen source. Right? If you are trying to culture out nitrosomonas, then depending on the environmental conditions, if you are trying to culture out organisms that can survive under high thermal conditions, under high heat conditions, you can increase the temperature and that will favor the growth of those organisms. It could be high salt conditions too. You can increase salt content within the media. You have media like mannitol salt agar, which we can use for isolation of our staphylococcus. These media are designed in such a way that their high salt content helps to increase salt conditions, salt concentration within the media, thereby making the media unfavorable for other organisms. So reduces the competition and allows the growth of organisms that can survive in such conditions. So sometimes it could be an issue of oxygen, you know, whether the presence of oxygen is favorable for the organism or not. We could use that to enrich our culture. So different types of you have nitrogen fixing bacteria, you have sulfur using sulfur bacteria, you have methanogens, you have all these different kinds of organisms which could make use of different types of conditions. And this is important for us to use. Now, why do we need to use enrichment cultures? We use enrichment cultures if we want to study biogeochemical cycles. So, our carbon cycles, our nitrogen cycles, our ion cycles, all these cycles have a lot of microorganisms playing a lot of roles. Nitrogen cycle have a lot of microorganisms playing a lot of role. And if we want to study the role of these microorganisms in all of these cycles, we can do so by enriching our culture and encouraging the growth of those particular microorganisms to the detriment of other microorganisms. When we do medical and clinical microbiology too, we're interested in particular organisms. For example, you're interested in organisms causing sexually transmitted infections. So you can take your time to, create conditions and cultures that encourage the growth of those organisms over some other kind of organisms. When we do environmental microbiology, when we do industrial microbiology, all of these are there. So it's important to understand this about enrichment cultures, that they are very, very important. But some of their disadvantages include that there can be overgrowth of the culture, which may not be for the best of what we want to do. If you have and overgrowth. And sometimes it leads to loss of diversity. Because the interesting thing about microorganisms is that they are always diverse in nature. You can see so many of them at the same time. So if you have a situation where there is a loss in diversity, that can affect, what it is that we are bringing out from our sample. Thank you for listening. Remember to like, remember to share our content, remember to love your science. Have a wonderful day.